I'm Jo Barnes from the University of Auckland in New Zealand, and I'm lead of the Western Pacific chapter of the International Society of Pharmacovigilance. My co-chair for this session is Dr. Mira Woolrich, current president of ISoP and also based in New Zealand. And Mira and I are delighted to be your co-chairs for this event. First, thank you to each of you for being here with us today. We acknowledge that these are challenging times, and we hope that all of you and your families are safe during this difficult time in the middle of the COVID-19 pandemic. The pandemic has certainly reinforced the importance of having robust evidence and information on the safety and efficacy of medicines and vaccines, the importance of good risk communication, and the interplay of these with patients' perceptions. And I'm sure that these topics will come up in our discussion time after tonight's presentations. So on behalf of the International Society of Pharmacovigilance, we are very pleased to welcome all of you to the second ISoP Patient Safety Day event that will be held this year over September 16th and 17th. By way of background, the World Health Organization established Patient Safety Day to help increase public engagement with safety issues in healthcare and to promote global action to prevent and reduce avoidable harm in healthcare. ISoP supports this idea to promote patient safety, particularly in relation to use of medicines, and has therefore included topics on the theme of medication safety for the Patient Safety Day sessions. We hope you will be joining some of the other sessions as well as this first very special one. For those of you who are active on social media, there are a few hashtags that you can use in relation to this event, and I think we have a slide later that will show these for you. But just for now, a couple of them are #ISoPPSD 2021 and #World Patient Safety Day. It's time now to get our programme underway. And to start, we have a short welcome video from our ISoP president, Dr. Mira Woolrich, who you have just seen. Jenny, could we play the video from Mira, please?
Welcome to this webinar organised by the International Society of Pharmacovigilance for World Patient Safety Day 2021. My name is Mira Woolrich and I'm the President of ISoP. Our professional society is a worldwide organisation with several hundred members working in pharmacovigilance. Our aim is to promote the safe use of pharmaceutical products in all countries. World Patient Safety Day was established by the WHO in 2019. And it's marked on the 17th of September each year. It's an important day for ISoP because it promotes patient safety as a global public health priority. Every year, millions of patients die from an adverse event, and many of these are related to pharmaceutical products. This year, with the continuing COVID-19 pandemic, global awareness of medicines and vaccine safety has been extremely high. The everyday work of those of us in pharmacovigilance has made international headlines on a regular basis. ISoP members work in very many different sectors, from drug development, medicines regulation, academia, to pharmaceutical companies. And for all of us, the end point of our work is patient safety. Every issue analysed and discussed is meaningless unless we consider how it impacts on the patient themselves using medicines in their own environment. Drug safety issues also affect the family and friends, including children and carers of patients using medicines. I'd encourage you to think about this broader holistic approach as you watch this webinar today. The WHO theme for World Patient Safety Day this year is maternal and newborn health. So please remember this because it is especially relevant. Wherever you are in the world, I hope you enjoy this webinar that we've made for World Patient Safety Day. We hope you find it interesting and informative. If you work in pharmacovigilance and you'd like to join ISoP, please have a look at our website, isoponline.org, and consider making a membership application. We would welcome you in joining our professional society so we can all work together towards improving patient safety worldwide.
Thank you, Mira, for that excellent video and for being here with us this evening to co-chair this session. It's really great to have you here. I would now like to introduce our first speaker, Professor Libby Ruffhead, who is going to discuss digital biomarkers and can they provide opportunities for improving pharmacovigilance. Libby is a National Health and Medical Research Council Senior Principal Research Fellow at the University of South Australia in Australia. Her expertise includes pharmacy, behavioural science, epidemiology and public policy, with a research focus on improving the quality use of medicines. Libby is a Fellow of the International Society of Pharmacoepidemiology and a Harkness Fellow in Healthcare Policy and Practice. She's Director of the Quality Use of Medicines and Pharmacy Research Centre and Executive Director of a national programme for the Australian Government Department of Veterans Affairs. Libby is currently a member of the Australian Government Drug Utilisation Subcommittee of the Pharmaceutical Benefits Advisory Committee, the Medication Safety Oversight Committee of the Australian Safety and Quality Commission, and the Medicine Safety Programme Steering Group of the Australian Digital Health Agency. So we're very honoured to have Lizzie speak with us this evening. Just before I ask Lizzie to begin her presentation, Libby, sorry, if members of the audience do have questions, if you could please put them in the chat pod or the Q&A, and we will have some of those questions put to our speakers in the discussion session after the two presentations. Welcome, Libby, and we look forward now to hearing your presentation. Thank you.
Thank you, Jo, and thank you, Mira. So today, I want to explain before we start that I'm no expert in digital biomarkers, but I'm just hoping that we can have a conversation about it. And what I want to kind of think that my thesis for this talk is that digital biomarkers are the next frontier in pharmacovigilance. And I think they will provide us with the potential for early identification of hard to detect harms at the patient level. However, there's not a lot of research in this space. And so I'm hoping you'll join with me in a discussion. So please put in lots of questions, but discussion for everybody on whether or not the use of digital biomarkers is worthy of an exploration by the pharmacovigilance community. And I'm going to touch on a little bit of the work that we've done in this space, but more about the potential in this space. But to kind of put us in the space of the next frontier, I'm going to start with a little bit of history. So I'll just try and get this off my screen so it's a bit easier for you to see all of my slides. Oopsie. But anyway, if we think about pharmacovigilance, I'll think about it from the Australian perspective, which is where I live. But it started in Australia in the 1960s and 70s. So in 1964, we started the Australian Drug Evaluation Committee was established. We established our spontaneous reporting system in 1970, and we set up an Adverse Drug Reactions Advisory Committee to analyse those. And our aim at that time was to identify previously unrecognised adverse events. It's worth thinking about what healthcare looked like at that time. I can't get data from the 60s and 70s in Australia, but I can get data from 85. And in Australia, most people had an acute illness or one chronic illness. We didn't have lots of people. This is the people here with two or more, but one or two, but we didn't have many people with multiple chronic illnesses. And if we treated illnesses, we treated it with one medicine. If I go forward to the 80s, 90s and 2000s, what we see in 86, drug safety, the journal began. ISoP, the European Society for Pharmacovigilance, began in 1992. Pharmacoepidemiology and drug safety, that journal began in 1992. And we got ISoP in the year 2000. Across the 90s and 2000s, as we computerised, we got large databases. And so for the first time, we were able to do observational studies of harms due to medicines. And I think the opportunity that this gave us was to go from, is there a side effect, to how much risk is there? And who are those groups most at risk? And just as pharmacovigilance changed, healthcare changed. So I showed you 85, now I'm moving forward to 2005, and this is where we see a real jump. In 85, we only had 10% of our population with 3 or more chronic illnesses, and by 2005, it was 39% or more. The same thing that happens as that time is we've got multiple pharmacotherapy emerging. So it was 1998 was the first time we had guidelines that said use 2 medicines for diabetes, not 1. And in 2000 was the first year globally that we said use 2 medicines for blood pressure, not 1. So multiple pharmacotherapy coming in at a time when we have multiple chronic illnesses. Jump forward to one more thing before I move on. That space has still got a lot of work that we need to do. There's still many drugs we don't know the risk about. We've still got to identify new side effects of medicines. And then there's all of this work to do just in reporting harms by age, by disease state, and by dose. A lot of that observational work we do with the large data sets, we seldom report the dose that was analysed when we identified the harm. We very seldom, while we've got better at doing numbers needed to harm, we seldom, if you could see this is the number needed to harms for opioids and hip fractures in the population 50 and above. But if we do it in 5-year age splits, you can see those numbers vary quite dramatically. And this is a similar thing. This is number of people we need to treat or cause, with NSAIDs to cause one NSAID-related hospital admission. And again, it varies by disease state. So there's still a lot of work to do in this space. But for me, the emerging space in the 21st century is the 2020s, when I want to say we're in the era of polypharmacy in a big way. And our big challenge is to improve health and minimise harm in people with multiple illnesses on multiple medicines. And you can see that in the publication of Medication Without Harm, the current global patient safety challenge. And this picture on my right is a report from the Australian Pharmaceutical Society of Australia on medication safety. They've been reporting annually on these topics, and Australia now has medication safety and quality use of medicines as its 10th national health priority area. So just to show you how quickly things have changed, this is data from the UK and it's 1995. This is the percent of the population. The colours here are the number of medicines people are on, none, 1, 2, 3, 4, and I need to get that away because you can't see the bottom. And lots of medicines. So, and across the bottom here is age. So in 1995, 35% of people who were 80 or more were on 5 or more medicines. Fast forward to 2010, so only 15 years later, and it's jumped dramatically. We've got 65% of people who are 80 or more on 5 or more medicines. Is the UK an outlier? No. This is data from the US, and in 1990 to 2000, which was the bottom, we've got 20% of the population on 5 or more medicines. And just 10 years later, 2010, we've got 40% of the population on 5 or more medicines. When I look at this data, I'm just staggered at how quickly this shift has happened. And of course, it's complicated. It's a complicated shift because this data is from the same study as the previous slide, and it's looking at how many unique medicine regimens do we have per 100 people who are taking a given number of medicines. And if you're elderly and on 5 or more medicines, there are 100 different unique drug regimens per 100 subjects. So this means guidelines and other things aren't really possible because polypharmacy regimens are unique. And of course, they interact. So again, this is showing you if you're on 10 or more medicines, total drug interaction alerts per person is 13.7. So more interactions than we have the medicines that people are taking. And of course, that leads to a lot of harm, and a lot of that harm is preventable. These are Australian data, but we estimate we have 250,000 hospital admissions per year that are medicine related. We have another 400,000 emergency room attendances. We have more than 1 million Australians suffering adverse medicine events every year. 1 in 5 older persons at any one time seem to have an adverse reaction if we're going for a medicine review. And it costs us $1.4 billion. This data on the right is just showing you the percentages of medication-related hospital admission in different groups for Australia. These are all the Australian studies on this topic. This is our population who are 65 and over, and all of these studies are done in the group who had unplanned hospital admissions. And what you can see is it's between 1 in 5 and 1 in 3 of all unplanned hospital admissions in the over 65s are medication related. And the blurbs at the top, or bubbles at the top, are from the latest 3 studies which have been done most recently, 21, 2019 and 2014, showing the preventability estimates. So lots of medicine use, lots of harm. And of course, multiple medicine use leading to more harm. So again, this is showing you the number of sedatives you're on and the number of falls you have had, or the number of anticholinergics you're on, and the likelihood of hospitalisation. For confusion, 1, it goes up, 2, it goes up more, 3 or 4, it goes up more, and 5 or more, it actually is up to 3.5-fold and up to 4-fold if you're on 3 or more anticholinergics. So lots of medicines, multiple medicines make it worse, lots of harm. And then, of course, we have this problem, the hard to recognise adverse reactions, which are often not recognised at all and usually misclassified as ageing. So what happens when you've got a little bit of cognitive impairment? You've got a little bit of muscle weakness or a little bit of inactivity. You're just sitting in the chair a little bit longer or falling asleep in the chair in the daytime. You've got some constipation, you've got some incontinence, you've got a little bit of breathing difficulty. Very often these are medicine-induced, but very often they're described, misdiagnosed or described as geriatric syndrome or frailty. And of course, we have lots of evidence that shows us that the number of medicines you take is associated with frailty. And this is, oops, just one example. I've gone too far. Oops, back. Need to go back. So, you know, this is, if you're on 9 or more medicines, your risk of the likelihood of being frail is much higher. So that, I put all of that information together because it was that kind of thinking that led us to this question, which you can't see, sorry about that. Can we reduce the harms from medicines by detecting medicine-related harm earlier? If we could, so this was the theoretical model that we kind of came up with, that medicines we know are associated with adverse events, medicines we know are associated with frailty, but there's these intermediary pathways. Medicines are affected by sedation, medicines affect sedation, gait, walking, balance, they affect appetite, they affect cognitive impairment. So we were calling this medicine-induced deterioration. So if we could pick this up, could we stop people taking the medicines that cause these problems, which would stop frailty and perhaps adverse events via this pathway, or stop adverse events via this pathway? And there was plenty of evidence to tell us that these intermediary pathways are a real problem. You know, that a 10% increase in sedentary time increases frailty risk by 55%. A 30-minute increase in sedentary time increases the rate of mobility disability by 10%, which is that inability to walk 400 metres in 15 minutes. Every extra hour you sit increases the risk of death by 19%. And if you're physically frail, then it affects your cognition, and we can see a 1.5 drop in cognition scores. But it's not all bad news. If you can take an extra, 500 steps a day, you'll reduce that risk of major mobility disability. So what tools would help? And I just wanted to just highlight where the current pharmacovigilance data doesn't always give us the information we need, because many of those side effects that I was talking about are medicines, confusion, sedation, or drowsiness. And we can ask the question really well, which medicines cause those side effects? And if I go to cider and I type in drowsiness, I get 301 medicines. And if I type in confusional state, I get a whopping 542. So we have clear idea of which medicines cause these side effects. And if I go to cider it'll tell me what frequency those side effects are likely to occur. So I know it's more common with bromazepam, perhaps at 32%, than it might be with atropine, where it's rare, but apomorphine at 35%. So, but what I don't know is how much sedation those different medicines cause. And I certainly have no comparative information on whether one is more sedative than the other for lots of these things. I can make a good guess, but I don't have that information in a quantitative way. So that led us to the question, would digital tools help us to detect medicine-induced deterioration? At the time we were pondering this question, there was research like this out, which was someone using accelerometry to measure frailty. And within that study, luckily for us, there was some work done on medicines. So they were measuring changes in walking speed, mean stride time, variation in stride time, step consistency. And I've discovered my smartphone tells me all of those things if I want to look at it. What they found was that, and the graphic I've got there is step consistency, but polypharmacy and antidepressants and anti-epileptics were significantly associated with these changes in step, strides, other things, as were other frailty markers. So we kind of thought, well, accelerometry might be one of the ways we can get to detect this medicine-induced deterioration. And then at the time we were contemplating this, there were many other studies that were looking at devices predictive of falls risk or frailty risk, and they're just a number of them. So that's what we thought we'd do. We'd trial the use of accelerometry as a part of a randomized controlled trial we were doing to stop medicine-induced deterioration. We wanted to have older people wearing a health professional grade accelerometer, which is what this is. They couldn't read it themselves, it had a closed face. And we wanted pharmacists to come in before, over time, and monitor changes in activity as medicine use changed. So just to show you what these devices can do, they measure time spent in sleep, sedentary time, light activity, moderate and vigorous activity. And here, these are reads from real patients. The dark blue here is sleep time. And if there's a kind of line across this, it's kind of disturbed sleep time. The orange is active time, light active time, and the light bluey colour is sedentary time. So here we've got a person who's got good sleep, good activity, and some sedentary time. Here we've got another person who's got quite a lot of dark blue in the sleep. So they're sleeping, but they've got very disturbed sleep, but they've got quite strong pockets of activity. And here we've got a poor person who doesn't seem to sleep at all, and you can see lots of sedentary time and not so much activity time. So, and if they do sleep, it's really disrupted sleep. So here was our aim, that the pharmacist would have a readout, they'd have another readout, and let me tell you that the pharmacists didn't have to read these things. They could look at them, but they also got this kind of activity which said how many bouts of moderate activity had you had that were longer than 2 minutes, how many steps per day, sleep duration. And so the idea was that pharmacists would look across time and be able to assess if these had changed, had also the medicines changed, and was this likely a side effect of the medicine? Having told you all of that, I'm really sad to say that our idea and that technology was ahead of the IT infrastructure in Australian aged care. And so the lack of Bluetooth connectivity in many of our nursing homes meant that we couldn't actually get the data uploaded to the cloud to everybody. However, we did collect baseline 6-month and 12-month data using a research grade device, and the research grade device didn't need Bluetooth connectivity because all of the data was stored in the device. The problem with the research grade device was that it had to be taken off people and brought back to the lab to have the data downloaded and to be recharged after a week. So we couldn't leave it on people continuously. But we did collect that data, and we did collect people's medicine use at baseline 6 months and 12 months. So we had a correlations between the medicine use and the 12-month, and it was all collected prospectively. And so this is what we found, and I'll just need to explain this graph to you, because how you analyse the data from the accelerometers is in a 24-hour movement day, because there's always a trade-off. If you sleep more, then you've got to trade something off. And so this green line here is change in sedentary behaviour. This purple line here is change in moderate or vigorous physical activity. This aquary coloured line is change in light physical activity. And this pinky coloured line is change in sleep. And here's our medication load, and the column, it's a nuisance having that thing in the way. Sedative load is the left-hand side and anticholinergic load is the right-hand side. And there's a bit of overlap between those medicines, as most of you will know. But the important thing is, and so here's change in activity, and this is minutes per day. And so here's an hour or 50 minutes change, 25 minutes change. So what we see here is that if someone goes from 2 sedative medicines up to 4, or not to 2, it's pretty much the same. If you change by 2, it's a 30-minute change in sedative time. It's a 30-minute increase per day every day. It's a decrease in moderate physical activity by 2 to 3 minutes. Oh, that's probably a bit more than that, 5 or 6 minutes a day, which when you're 80, so this was all collected in people in aged care facilities. These are bigger amounts than it might seem, and light activity goes down and sleep gets worse. So much more disrupted sleep. So, and this is clinically significant. You'll remember I showed you this, that every 30 minutes decreases the ability to walk. 10% increase in sedentary time does increase that risk of frailty. So, and of course, it increases that risk of death. So we're pretty pleased with this, I have to say, because I do think gives us a way of thinking that there just might be other ways to measure harms. Like if we could get a sense that people's physical activity or sedentary behaviour had changed quite dramatically, maybe we would be able to change that medicine before it led to the catastrophic fall where they broke their hip. Or if we couldn't trade off the medicine, maybe we'd think, could there be something else we do? Or maybe it would stop that cumulative load, that we put one in and we take another off, so that kind of de-prescribing work. But here we have some objective measures to think about it. So I told you I wasn't an expert in digital biomarkers at the start, but this is where we started to, and this is where we've currently got to. But I think for us as a community, it's worth thinking about the opportunities. You know, smartphones, we've already talked about smartphones and steps, but smartphones and cognition, taps and swipes and all sorts of other things. This is a small study from 2018 and done in young people, but working memory, executive function, language and intelligence, they could all be replaced from neurological tests, or not perhaps replaced, but a correlation between the neurological tests and the predicted scores from you tapping or swiping your phone. That work's moved on quite a lot, and this is a very complicated graphic, but it's just showing all the different kinds of functional neurocognitive tests you might be interested in, and the devices. And essentially, because smartphones now have accelerometers, gyroscopes, GPS, microphones, cameras, timers, screen interactions, metadata, they're all considered now useful perhaps for the purposes of training, monitoring, diagnosis or rehabilitation. It's worth remembering that lots of technology is just rolling out as we speak. Falls detectors, people are wearing them. This one's one that goes on the floor in the room for when you get out of bed. This one's an in the mattress one where it's a pressure visualization, but it's also, it identifies falls. This is another one that's in the room, in the bed, sorry, but it's also detecting sleep apnea. This one, fancy one, in mattress device, tracking heart rate and respiratory data. This one for babies, tracking breathing. Accelerometry, moving from bracelets to rings, and even to hearing aids. This one I found really quite interesting. This hearing aid will do activity tracking, falls detection, and engagement tracking. It's actually testing how much you're engaging with people, because how much are you hearing and how much are you engaging? And then, of course, we have vital sign measures. We have Alexa and Google Assist, so the passive in-room sensors that can detect how active people are, devices for urinary incontinence. They're emerging everywhere. They're coming into life in all sorts of places. Oh, one more I found, meditation. You wear this one around your forehead. But again, mind, heart, body, breath, all of these things that these devices are currently checking. So for me, the opportunity here is really big if we can think about it. We've got continuous digital data collection happening in very small epochs of time. We've got medicine exposure and unexposed periods, medicine re-exposure, and additive medicines. So I think we've got the possibility of using this at the personal level, you know, an N of 1 study, and you know, re-challenge and challenge might be possible in ways we haven't thought about before, because we're able to perhaps detect the beginning of a deterioration event, not the catastrophic fall or the catastrophic outcome. These kinds of exposures allow us to think about how self-controlled designs would be work. And of course, the temporal effects hopefully are more easily able to be observed, but the added advantage of cumulative exposure. But not just for the person, also for the organisation and also at the population level. As I was preparing this talk, I kind of put in, you know, wearables and adverse medicine events or adverse drug events into search. I didn't really find many, you know, this is an emerging space. We are beginning to see wearable devices and, you know, what's the impact on major adverse events following lower extremity. We're certainly seeing the diagnosis kind of stuff. Here are ones about home-based monitoring, saying the passive centres have the best amount of evidence at the moment. And here's one of the few I found that was actually trying to do continuous monitoring of an activity tracker by patients undergoing chemotherapy for urothelial carcinoma. And it was trying to look at the side effects of the chemotherapy, and it was used fatigue and Fitbit as one of the effectives. But it's a research lacking space. So I'm going to conclude there, but by saying, you know, digital technologies are expanding across healthcare, and they offer this continuous data collection. And for many of those hard to identify conditions, I think they give us a wealth of information. And many devices are being created for monitoring the health of the elderly. So those precursor symptoms, I think this is where it offers us some opportunity, but we're not yet harvesting that information. I'm not a great advocate of asking people to wear this specifically for us, although maybe in clinical trials we might think of some of that stuff, but certainly the existing data that's just being rolled out in routine care, particularly aged care and hospital, I think offers a really amazing opportunity, but we're not yet there. Like, we need a lot more research to identify which tools work best. We need a lot more research to say what are the minimum clinically important differences for these precursor symptoms, and then a lot more research to think about how we might integrate and bring that into practice. So I'll stop there and come back and see you all, but some thoughts from me.
Thank you so much, Libby. That was a really excellent presentation, and it's so fascinating to hear you talk about this as a new frontier. So thank you so much for sharing those thoughts and insights with us. It's been a really fascinating and stimulating presentation. Thank you. I think we can take 1 or 2 questions just now, and then the rest of the questions we'll try and accommodate those in the discussion session later. So just while we're digging those out of the Q&A and the chat function, I just had a quick question, which you've touched on really, but hadn't quite said this word. But I was just thinking in terms of the types of digital technologies. I know that personalized robots, you know, are sort of being used in some contexts for the elderly in terms of assisting with medicines adherence and so forth. Would you see any scope for a personalized robot in a person's home that might in some way be able to detect or monitor potential harms or be used in some way to prevent harm?
Look, there's plenty of people who are more expert than this than I am and have thought about it more, but certainly my understanding is that we know that the in-room passive sensors can be used for detecting changes in people's kind of healthcare at some level. If people have deteriorated, they're moving around the house less, they're doing less activities and those sorts of things. And that's what I mean about here's the potential of this space. It's moving really fast. There's lots of things happening in the space. And that's why I'm kind of saying to us as a gang, can we think about how we actually engage in this space? Because I think it could do a lot, but I'm no expert.
Okay, thank you. Thank you, Libby. And probably the next two are a little bit along that. We have one question asking, which challenges do you think pharmacovigilance will be facing regarding these new digital technologies? And also, are these biomarkers precise and accurate?
That's why I said we need some research. Were any biochemical biomarkers measured? I haven't seen any biochemical biomarkers, but that's why I think we need the research. Look, for me, I think it's the stuff, for me, the stuff that would be most important that we do is the stuff that matters to patients every day, which I think is the stuff that's neglected. I think the impact of constipation from medicines, the impact of urinary incontinence, the impact of, things like, like little mild anemia, you can't get your words out as fast enough, and no one says it's a side effect, but of course, if you change your medicines, you could find and remember everybody's name. Like, this is the stuff I think we neglect, and I think this is the opportunity where medicines have an impact on your everyday quality of life, but not a severe enough impact to kind of make you say it's illness, but for people it is, so I think for me that's the priority space, I think.
Thank you, Libby. That's really excellent. I think we'll save the rest of the questions for the discussion, and I'll hand over to Mira now to introduce our next speaker, but hopefully you'll be able to stay with us, Libby, and join us in the discussion shortly. Thank you so much for your excellent presentation.
Thanks, Jo. And thanks, Libby, for that great presentation from me. And thanks to everyone who's attending this evening. I have been keeping an eye on the participants gathering, and I see lots of friends and colleagues from our Western Pacific chapter. So welcome to all of you. I can't see you, but hopefully you can see us okay. I would like to thank the ongoing help of Jenny and her team for organising this webinar this evening, and to Jo for chairing our Western Pacific chapter. So now, moving on, it's my very great pleasure to introduce Professor Ian Wong to deliver his talk to us tonight, which is called COVID Vaccine Adverse Event Response and Evaluation Programme. Many of you will know Ian. He's very well known to us in ISoP. He's quite a famous pharmacovigilante who was actually a former vice president of our society. His credentials are very impressive. He's a professor of pharmacy at University of Hong Kong. He is also a professor of pharmacy at University College London. He holds a joint appointment, and he's been a specialist advisor in England to NICE, the National Institute for Health and Care Excellence. And importantly for our region, he is a current member of the advisory committee to the National Prescribing Service MedicineWise database in Australia. So we're very honoured to have you with us this evening, Ian, to present to us. Your talk looks as if it's going to be most interesting. And I will now hand over to you with just one thing to say to everyone. If you do have any questions, please do type them in as we're going along, and we'll do our best to answer them at the end. Over to you, Ian.
Thank you very much, our President. It's so good to see you again. And okay, so I'm going to do my presentation. I'm going to share my screen. And okay, is it okay for the screen? You're seeing the presentation? Lovely, thank you. Okay, so thank you very much for the invitation. So I'm going to talk about this particular program, CARE program, which is a program that funded by the Hong Kong government looking into the COVID-19 vaccine adverse events so that we can evaluate it and also respond to it. So I'm going to give you the acknowledgement and disclosure and then tell you something about the background of the COVID-19 vaccine pharmacovigilance in Hong Kong. And also specifically, I'm going to give you a study that we have just completed and published it. And so that you can have a look how we use different methods of pharmacovigilance to evaluate the COVID-19 vaccine safety. So give you the acknowledgement, the project itself is funded by the Food and Health Bureau of the government of Hong Kong. And also we have opportunity to have the colleagues from our drug office in Department of Health, which is our regulatory authority, and also the hospital authority, which is the NHS in the UK. So they look after all the and I'm a member of, and also thank you the members of the expert committee. They gave us the data when they did the causality assessment, and also all the co-investigators from the universities in Hong Kong, and also my co-investigator and all the research team member. I have been a principal investigator or a co-investigator on different grants from different countries, different places, specifically from industry, but none of those grants actually related to the current study, except what I declared already. I'm a founder and a shareholder of a company called Ferrocan. It's a spin-off company in UCL. And also I'm a director of a healthcare innovation technology service and an executive director of a listed company in Hong Kong. And I just mentioned earlier on that I'm an advisor to different places, but again, it's the study itself organization. Okay, so give you some background. In Hong Kong, we have the prevention and control of disease, use of vaccine regulation. It allows emergency use of COVID-19 vaccine in Hong Kong. And indeed, I think most countries use the emergency, allow the emergency use to not the regular license, but a license to allow the emergency use so that we can collect more data. And within the regulation itself, it gives the provision of allow us to monitor the safety. And also Hong Kong government have set up a compensation fund in case any unexpected adverse event so severe affect the patient, then the patient can actually seek compensation. So you can see from this point of view why it's so important to have the pharmacovigilance program in place to monitor the safety of the vaccine and also to provide the information to the government to understand how to actually deal with any subsequent claim. So we have this particular program looking into two particular vaccine. One is Coronavac, it's from the Sinovac BioNTech. And also the other one is the Comirnaty, which is the BioNTech one, but it's actually marketed by Fosun, which is equivalent to the Pfizer BioNTech vaccine. So the CARE program is part of the enhanced pharmacovigilance plan developed by the Department of Health, but led by the University of Hong Kong by our team. It's a 3-year program and start with the COVID-19 vaccine program in February 2021. So the program been started for 7 months already. So the government specifically take reference to the European Medicine Agency Access Program. We're looking into 40 different conditions, which is used by the WHO Global Advisory Committee on Vaccine Safety. They include Bell palsy, Guillain-Barré syndrome, heart failure. So it's a whole range of what we call the AESI, adverse event of special interest. It doesn't mean that vaccine will cause this type of adverse events. But based on our understanding of the past experience of vaccine and also potentially to do with the COVID-19 infection itself and some of the pharmacology side of it, this list came out so that we can proactively monitor this type of adverse event. So that's why I call AESI, special interest. So for the study in Hong Kong, it costs about $1.3 million to run this particular program. So we actually have 4 different parts. So the first one, we have the background rate of adverse events of special interest. So we monitored for the last 10 years of this type of adverse event from our hospital authority medical record. And I'll explain a little bit about the hospital authority later on. But basically, it's the electronic medical record linked up with all the hospitals in Hong Kong. And also we have the active surveillance of AESI, adverse events with special interest. So we use the electronic medical record to do the active surveillance. So every week we will screen any patient they admitted to hospital with this type of specific AESI. And then at part 3, we do the evaluation of the safety of COVID-19 using electronic health record. We do the record linkage study. We link the hospital authority medical record with the Department of Health immunization record. And then part 4 is intensive monitoring study. We recruit subjects to conduct a prospective study looking into a relatively common adverse effects such as fever, pain in the injection site, dizziness, this type of adverse event. They are not going to be the one that will cause significant harm, but we want to have a comprehensive risk and benefit assessment. And that's why we recruit about 1,000 patients on each particular vaccine and follow them up for 28 days. But today I want to specifically looking into the part 3 study to evaluate the safety of the COVID-19 vaccine using record linkage study. To give you a bit of the introduction of the Hong Kong healthcare system, the Hong Kong healthcare system been successfully ranked into at least the top 3 in the world. Is the cost of the cost effectiveness of the system. We've been ranking as one of the most efficient system in the world. The characteristic of the Hong Kong healthcare system, in terms of efficiency, we have about 95% of Chinese, and we have a universal coverage of the public health system, heavily subsidized by the taxpayer, and most of the chronic illnesses and severe illnesses are treated by the public healthcare system. And the private healthcare system, we have very large number of private primary care physicians in Hong Kong, but they tend to look after minor ailments like cold, flu, fever, but they don't tend to look after the chronic illnesses. And the private hospital look after people with insurance or relatively well-off citizens. But because of the public health system is so efficient and also so cost-effective, there are a huge number of people are treated by them. So our public healthcare system mainly run by or completely solely the public-funded healthcare system is hospital authority, which was modeled by the NHS system in the UK. Hong Kong used to be the colony of United Kingdoms. So you can understand why we have quite a similar system of the universal healthcare system provided for every single citizen in Hong Kong. The hospital authority, the HA, is serving a population over 7 million through 42 hospitals, 47 specialist outpatient clinics, and 73 general outpatient clinics. We are heavily subsidized, very, very popular, highly popular. Give you just one example. If you require H2 antagonists for your GI problem, for 3 months treatment, it costs you less than $2 American for 3 months treatment. So you can understand why most people, when they need chronic treatment, they would use the hospital authority system. The Hong Kong public health care system, we have a very unique situation. We have a unique electronic patient record in Hong Kong. And it's a single unit medical record. No matter which hospital you are in, you see exactly the same record, and all the data accumulate in a central system for all 42 hospitals, authority hospitals, and all the outpatient clinic. And they store in one single system for patient management. And they transfer the data to clinical data analysis and reporting system called CDERS. So it's for audit and research purpose. So this gives us a very unique opportunity. As long as you use the public health system for your treatment, we will have your data. And the CEDAR contains the data of diagnosis, procedures, drug prescribing, and drug dispensing. Now, we are also quite unique because the prescription from the hospital authority will be dispensed outside the hospital authority's system. So that's why we also have the dispensing data. We have the admission data, discharge data, laboratory and pathology results. We have another very unique link is the mother and baby link. Each child is born will be linked with the mother's record permanently. And currently it contains 11 million patient data because it's been around for 20 odd years. So it's more than 7 million patients that we have in Hong Kong at this particular point. So what I want to also look specifically into is the safety of the COVID-19 vaccine, and specifically looking into Bell's palsy. Now, we are authorized by the government to use the data for the pharmacovigilance. We have the IRB approval from both hospital authority and also from the Department of Health. Now, one thing is very unique, it may be different from other countries. When the new emergency law was implemented, it also authorized the consent from the vaccine recipient, not just the consent of using the vaccine, but also consent to allow us to use the data for safety monitoring. So effectively, we have the consent of every single vaccine recipient to allow us to access their medical record in order to do the pharmacovigilance. So we obtain the data from the hospital authority, and also we link the data with the Department of Health vaccination data, because the Department of Health is actually the vaccine provider, and they have electronic medical records to allow us to link them using the identified anonymized data. And then we, as the research group from the University of Hong Kong, we performed the epidemiological study. We disseminate the result to both general public and also peer-reviewed journals. Now, one thing I want to emphasize, the Hong Kong government is very transparent. They will allow the general public to know that we have regular update to the general public of the report of the AESI or any adverse drug reaction, adverse event related with the COVID-19 vaccine, and they allow us to also announce our data so that people will know precisely what we got. And that is quite unique, is to have such an emphasis of the transparency. So one of the interesting things is when we commenced the vaccination program until up to the May 2021, we have about 450,000 subjects, about 500,000 subjects on CoronaVax, and also for the biotech ones. But we already received 53 cases of Bell's palsy, and that really giving us a positive signal. We need to investigate it. And so basically the regulator, the Department of Health, together with the expert committee on the safety of vaccine, realized that we have so many cases within such a short period of time. And that provides us a positive signal for us to do the investigation. So the CARE program initiate a rapid evaluation of the study. So just give you a little bit of the Bell palsy itself. But this particular patient actually is one of the subjects have the Bell palsy after the COVID-19 vaccine. Now he actually wanted himself go to the newspaper. So he gave the perfect consent to the newspaper in order to take the photograph. And if you want to, you can actually click on the web link. You can even see his own video on how to present the Bell palsy. The Bell palsy have sudden onset of one side face muscle weakness, and also affect both upper and the lower face. Other symptoms may include numbness of the face, neck, or ear pain, and change in taste and sensitive to sound. It's highly important, it's only one face. So this is what you need to remember, it's not both faces. The exact cause of the Bell palsy is uncertain. And most scientists believe is that it's reactivation of existing dormant viral infection like herpes virus may cause Bell palsy. And also we believe that impaired immunity from the stress sleep deprivation, concurrent acute illness, or autoimmune syndrome have been suggested to be the most likely triggers. And indeed, we know that a lot of different vaccines have been suggested to associated with the Bell palsy. And the facial nerve, basically 7 cranial nerve, swells and becomes inflamed, leading to temporary facial weaknesses of one side of the face. And prognosis, Bell's palsy is usually transient, and the prognosis is generally very good. About 70% of cases will experience fully recover even without treatment. But if you have timely oral steroid treatment, within 3 days of symptoms could improve, and the chance of recovery will be more than 90% by 9 months. And physiotherapy is commonly prescribed with the oral antiviral agents occasionally prescribed, but of course the steroid is still the main treatment. Mild residual facial weakness, and the remainder may have moderate to severe deficit, but the outcome is usually excellent. But I must say that it is very scary when you suddenly have half side of your face weakness or sometimes even paralyzed, and you can imagine how terrifying to the subjects themselves. So I'm going to actually describe the study. We have 3 different sessions of the study. We're looking at the case report, and we compare the incident rate of the vaccinated people compared to the previous historical record. And also finally, we did the nested case controls study to looking into the association. So we go look into the case report reported to the Department of Health through the spontaneous reporting system. So after the assessment by the expert committee, 9 out of the 53 cases were reclassified as not Bell palsy. They were caused by stroke or varicella zoster virus, i.e. Ramsay-Hunt syndrome. So they are actually not caused by the Bell palsy. Bell palsy is supposed to be unknown orient. And so it's one of the things that why a lot of times that people have Bell palsy, we will send them to have an MRI or CT to exclude the possibility of a stroke, because that is the last thing that you want to have a misdiagnosis. Bell palsy doesn't usually cause any significant harm, but stroke do. And the Ramsay-Hunt syndrome is usually occur a few days later. They can see the blister come out, the virus, and then usually they get reclassified. So just looking into the data in here, you know that actually a lot of Bell palsy are not really Bell palsy, and that's why it's so important to still use the classical causality assessment, looking into the cases. Within these 28 cases, we can see that there are 28 cases in the coronavax. This is the we have a more male, but we don't know whether it is really male is more likely. But this is give you the understanding of the patient. Majority of them occur within 21 days after vaccination, and this is for the BioNTech one. So the BioNTech one have less cases, but it may be to do with because the BioNTech was the sort of like the second. We have the license from the CoronaVac to start with, and then the BioNTech was a month later to have the vaccination program. So maybe all to do with the number of the cases lower is all to do with the sequence of the vaccination program in Hong Kong. And again, all 100% of the cases are below 21 days after the 21 days after the vaccination. So they're quite close to the vaccination time. And also, after we exclude all those cases, they were not Bell palsy. We did the causality assessment using the WHO as assessment. In 2 cases, we have inadequate information, so we classify them as D, i.e., we don't have enough information to do classification. For another 42 cases, we classify B. We call it indeterminate. But the reason why it's indeterminate, because before this particular study, there were no other study ever been done to looking into the association. And as we know, a lot of causality assessment is based on our understanding of the previous study. But because there were no other study before, so we couldn't quite actually say that it's fit into what we understand or consistent what we understand in the past. since this is the first study, and that's why all of them became indeterminate. So that's why it's so important. After you do all the case review, we realize it. We need to do even more systematic. We cannot just rely on the case report or the spontaneous report. So we move into the second study. But this is to give you the distribution of the time, and you can see that majority actually occur during the early period of time, and then the second week. But then after the second week, we hardly have any cases. So now we move into the second part of the study, looking at the incident rates. We estimate the differences of the incident rate of the Bell palsy among the vaccinated subjects compared to the historical population rate. So we're looking into here of the total of about 776,000 doses administered between the 23rd of February to May 4. So we estimate the incident is around 3.61 cases out of 100,000 doses. And then we do the standardized incident rate will be 66.9 per 100,000 person years. But the person years one is actually very difficult to interpret because, as we know, most of the Bell palsy occur during the early period of the post-vaccination. So sometimes it's actually more helpful to just looking into per 100,000 doses. And indeed, you're looking into most of the literature, they do actually use the 100,000 doses as the denominator because it's easier to compare with different studies. So when we're looking at the standardized ratio using the patient data, so we can see that the age standardized background incident rate in 2020, using the 2020, we would actually estimate the coronavax would be about 25 per 100,000 person year. This is using the background incident rate. This is what you expect to see. But then when we're looking into what we observe, which is the second column, is the age standardized incident rate of the vaccinated group, we actually observe 66.9. So by doing it, we can actually calculate the difference. We observe extra 41 cases out of 100,000 person years. So the age standardized rate ratio is around 2.64. So by doing this one, if we fundamentally, the take-home message, we're looking into the incident rate of the vaccinated subject compared to the background incident rate of the 2020, we see about 2.6 times increase. So that is the bottom line. So now we're looking into the BioNTech one. And again, for the BioNTech one, calculate the incident using the 100,000 dose and also using the 100,000 person years. That gives us the figure again. In here is not a really huge increase. The background incident rate is not really high, but it's when we're looking into the background incident rate, and again we see the increase of about 1.66 times. So that is what you expect to see if the same population without the vaccination using the background incident. But if we looking into the observed, this is the observed rate is 40.8 per 100,000 person years, then we have a difference of 17 cases per 100,000 years. So that gives us the 1.66. Now, but it's not significant in terms of the confidence interval. So this is a graphical representation. You can use the graph in here that it clearly demonstrates there's an increase of the incident rate after you have the vaccination. So, but again, I want to say that the for the commonality or for the BNT162B2, the BioNTech one, the 95% confidence interval actually overlap with the background incident. But certainly that in here, it gives us the idea we are likely to deal with association between the COVID-19 vaccine and also the Bell's palsy. So that's from the second part of the study, we can conclude that we can see that there's a higher background incident rate in coronavac, but it's also slightly higher than the BioNTech one. And based on about information, the care program got to do the final analytical study by using the case control study. So now I'm going to look into the next case control study. So we identify all patients with newly diagnosed Bell palsy since the vaccination program. So they are our cases. And of course, as you know, like any other case control study, there should be someone have received the vaccination and some without. Similarly, we identify the control. The control are the people did not have the Bell palsy. And again, they will have some patients receive the COVID-19 vaccine and some without. Now, if the Bell palsy is associated with the COVID-19 vaccine, then the case control study should demonstrate more people have received the COVID-19 vaccine in the cases compared to the control group. And that is the basic principle of the case control study. So this is the flowchart of how we do it. So we identify people with the newly diagnosed Bell palsy between the period of time in the emergency room or in inpatient. So there were 328 patients. And then we use the control will be 363,000 patients. They were patients without the Bell palsy. And then we exclude patients that they're under 12 years old because there was no one actually immunized under 12 years old. And then we always have a little bit of the hiccup of missing this gender, missing the day of birth. Those have to be removed. And then we match them with 1 to 4 control with the age, sex, setting, and plus and minus 3 days. And then we excluded people that we cannot determine the vaccination status and excluded people they subsequently had stroke and Ramsay-Hunt syndrome. So they are likely not the Bell palsy. And then we're looking into the, and also the control. If they remove the case, we have to remove the control at the same time. And also, so eventually we have the 298 cases and then 1,181 control. And then we're looking into the baseline information. There are some differences, but it's not that we will have serious concern. I'm not going to go through it because it's quite tedious. But what we are going to show is, in terms of the Bell palsy, we can see that after we adjust of the baseline disease, we can see that the coronavac is actually have the adjusted odd ratio. It's about 2.39, which is very similar to what we demonstrated earlier on when we compare the exposure group with the background incident. We have 2 point something of the IRR, and now we have adjusted odd ratio of 2.39. So it is very consistent. And similarly, it's also very consistent with the BioNTech vaccine. Again, it's 1.7, 1.8, very close to what we detect in the early on using the historical incident rate. But again, the 95% confidence interval do not give us the confidence interval that to say that it's statistical significant. So from looking into the previous publication, there was a commentary in Lancet Infectious Disease do actually say that indeed in the clinical trial of the mRNA study, the estimated or the observed Bell palsy is between 1.3 to 3 times higher than expected in general public. If you're looking into what we provide in our case control study and also comparison with the historical data, it is giving you the similar range. And although the FDA basically say that there was no evidence that the BioNTech or the mRNA vaccine, including the Moderna one, have the Bell palsy as the adverse drug reaction. However, they do actually say that they need to, the company need to monitor it. And indeed, if you're looking into the summary of product characteristic, the product monograph is actually both BioNTech and the Moderna one are actually listed Bell palsy as the potential side effect, but it wasn't in the Janssen or AstraZeneca, nor in Sinovac or Sinopharm vaccine. And if you look into WHO veg-based database, what another group did the study published in JAMA Internal Medicine, they basically say that the reporting rate of the Bell palsy in mRNA vaccine is no greater than other viral vaccine or flu vaccine. Now that becomes very problematic because they compare with the other vaccine. And then we know that other vaccine is apparently there may be some association with the other vaccine with the COVID, with the Bell palsy. So if you say that you know higher than them, it doesn't mean that it doesn't cause the Bell palsy. And also there was a Israel study that have a very small case control study having 37 cases and 74 controls. And then they said that they didn't detect significant risk. But when we actually looking at this particular study, the sample size was only sufficient to detect an odd ratio of 4. So looking into our study, we already demonstrate the odd ratio is only about 2. So if this particular study was just not enough power to detect a difference of the odd ratio of 2. So that is one of the problems in a lot of pharmacovigilance studies. They just simply underpower, and then they give people the wrong reassurance of the drugs don't have the adverse effect. So I just demonstrated that our study is actually 10 times bigger than the Israelite study. So we have sufficient power to detect a difference. But I also want to point out the study was still too small to detect 1.7, which is what the ratio of the BioNTech one. And that may be one of the reasons why we didn't actually detect a significant difference. But you can see that the data do show a trend of increased risk. But what is important is from the regulatory point of view or from the pharmacovigilance point of view, there's no published data on the association of Bell palsy on the coronavac. So our study is actually the first study. And the implication is for the BioNTech one, their package insert or what we call the package insert of the patient information leaflet or the product characteristic summary already say that there will be potential adverse effect of Bell palsy. So from the regulatory point of view, the Department of Health in Hong Kong do not require too specific action except to educate the patient and the healthcare professional. There is a possibility of the BioNTech one causing the Bell palsy. But the coronavac, because it's not listed Bell palsy as one of the potential adverse effects, so there was after communication with the company changed the product insert to include the Bell palsy. And then subsequently, for the regulator themselves, they need to reclassify the WHO classification. Because remember, I earlier on mentioned that when we did the causality assessment, there was no other study ever been done. So we couldn't actually classify the study as consistent. We can only classify the cases as indeterminate. But now we have the study to demonstrate the potential association. So this is from the website of the Department of Health in Hong Kong. We see that they put down Bell palsy as a very rare effect. They put down observed post-authorization in Hong Kong, which is our study. And but the most important bit is we know that the Bell palsy is very rare. It's very transient. We estimate that it's only 4.8 extra cases out of every 100,000 coronavac vaccine recipient. And in general, 90% of them will completely recover. So the benefit, the protective effect of the COVID-19 vaccine far outweighed the risk of this general cell-limiting adverse event. And the paper was published in Lancet Infectious Disease on 17th of August last month. And of course, there was a commentary actually come with it. They actually agree with our conclusion earlier on. The benefit is far outweighed the risk. And so we should encourage people to continue to use it. And indeed, a press release came out from the Lancet Press Center, and they agree totally about the Bell palsy benefit, the COVID-19 vaccine benefit far outweigh very rare risk of Bell's palsy. So the care program continue to work on different aspects. We are currently working on the myocarditis and pericarditis, thromboembolic events, seizure, blood dysplasia, autoimmune, so and so forth. And I think the care program actually demonstrate the full range of methodology can be used on pharmacovigilance when you have a very powerful record linkage system. And we are actually PM a little bit at this particular point. We just finished a self-control case series study looking into the Bell palsy associated with the coronavac vaccine. And now we accumulated far more data. We just finished a study with the case controls and also the self-control case series of the Bell palsy. And now we have sufficient power to detect the difference. So watch the space. You should be able to see our next paper very soon. So thank you very much. And hopefully we will be able to give you more information. So feel free to go to our website. In here, you will be able to link up with all the study that we conduct. And thank you for your attention. I'm happy to answer some questions.
Thank you, Ian. Thank you so much. I feel like clapping for all of us because that was an excellent presentation. And I think the first thing I want to say is just how it illustrates what impressive systems you have in Hong Kong, both health systems and systems for data collection, which have allowed you in your work to move far beyond spontaneous reporting, which of course is the cornerstone of pharmacovigilance, towards what many of us have wanted to do for so many years and move towards intensive pharmacovigilance, where we have denominator data, where we can perform pharmacoepidemiological studies as you've done. And in this impressive work on Bell's palsy, you've done 3 types of study all in one, and you've been able to publish a very strong paper in a very high quality journal. So congratulations on that. And we got a lot of comments coming through the chat. I could see while you were talking about how impressive the systems are in Hong Kong. I've just got a question myself. Joe will come in a minute with some more questions that have been coming through on the chat. But could I just ask you, you mentioned quite interestingly when people are vaccinated that you not only gain consent for the vaccination, but also for to use their data and access their health records. Is that a verbal consent or is it a written consent?
It is an electronic consent because what happened is in Hong Kong have a website, people got to book the vaccination time and also choose what they want. They can choose whether they want to have the coronavac or the BioNTech one. And so that's why when they're looking into the whole things, they already given the consent and they click on booking it. We did actually seek the advice from the Ministry of Justice. To see that whether this is legal and clearly that the verdict is legal. The reason is because the vaccine is actually under emergency use. It's not a regular medicine. It's not the long mode license. So the government is actually taking the responsibility of if anything goes wrong, they will take the responsibility. So they should have the right to actually monitor.
Right, that's interesting because I sit on the National Ethics Committee in New Zealand and we address these issues of consent for access to health records and data, but it sounds as though it's been covered by this emergency use. I just wonder, is there anybody, have you got any figures for the number of people who don't consent to their data being used and their records being accessed?
We don't know that because when they consent for the vaccination, it's a bundle together. So they have to consent to allow us to actually monitor. Yeah. So that's why and also this is getting extremely challenging if you try to actually decouple the situation, because the provider of the vaccine, the Department of Health, they're also responsible to ensure if anything goes wrong, they can actually take the responsibility. If they don't have your consent, they're not able to actually to maintain their ability to protect your health in a sense.
Yeah. Another question that came through on the chats relating to this is somebody asked who can access the data once you've collected this data on these cohorts. Who can access these databases for research purposes? Does it follow the usual guidelines for health research, or can you tell us a bit more about how that works in Hong Kong?
Well, for this particular study, it's very unique because it's under the emergency law to do that. And in general, for other research, so basically, I just talk about in general for using the database in Hong Kong. We don't usually need to use the Department of Health database because the Department of Health database is mainly about the vaccination record. Let me give you an example that we just published a paper in JAMA Internal Medicine looking into mother exposure to medicine and then the outcome of the baby. So we only need the data from the hospital authority. For this type of situation, we get the IRB approval to start with. And then secondly, we request the data from the hospital authority and to make sure that the data that we have will not be able to reverse engineer to identify who are the patient, who are the baby. So they are the anonymized data. And only the academia in Hong Kong will have the access right to this type of anonymized data. So because at the moment, the system is still, we are probably sort of like the first few group of people using the data to such an extent. So at the moment, it's not open up to the people beyond the border at this particular point. But we do work with other groups. Indeed, I work with Joe, I work with your group in New Zealand, looking into international comparisons of like fracture or use of ADHD medicine. Then we are able to actually work with overseas collaborator, but it got to have a Hong Kong local investigator to drive the data, to use the data in Hong Kong.
I wonder if we could just bring Libby into the discussion with a related question relating to your presentation, Libby. There was a question around whether there are any ethical or privacy issues with continuous data collection from older people. You just unmute.
You just need to unmute yourself, Libby. Can't hear you.
Yeah, look, always when it's being used for research purposes, you must obtain consent. I wouldn't be advocating at all that we're kind of using this data without consent. But I think that's I think they're the ongoing conversations to have. How do we help people thrive, and how do we use these devices and do that in an ethical way? But certainly, yes, there's lots of consent issues to think about.
Yeah.
Did the older patients in your study, Libby, do you think they understood, they all had competency to consent to and understand how their data was? Because I think to a lot of people it would be quite scary, this idea of a continuous data collection of their personal information. Where's it going and who's seeing it?
So this was a really interesting kind of thing. So because we were trying to stop deterioration, and I didn't say those things, we excluded people who couldn't consent, so people with dementia weren't in. But one of the interesting things for lots of people wanted to know, they'd never seen this kind of data about themselves before, so they wanted to know their activity. But the other kind of thing that was useful, we were doing some other stuff like cognitive tests, and they loved it because they'd say, my family have been telling me I'm forgetful and you can tell me I'm not. So sometimes that kind of, when family members are worrying or other. So it has, I think it's both of those things, but interestingly, this is creeping in without consent. Like all of these devices are being rolled into nursing homes. That information is available to the facility and it's probably being collected by the manufacturers. How often does the false alarm go off? So it's being collected without consent, I think, without us even realising.
Yeah,
and on World Patient Safety Day, which these events are for, I think the reason I'm raising these issues is to think about the patient or the participant in a study, to see things from their point of view, how their data are handled. But it also brings me on to another issue which Ian and I have discussed in pharmacovigilance circles, which is about misinterpretation and misuse of pharmacovigilance data. And I just wanted to ask Ian if he had any comment to make, because we've seen with the monitoring of the COVID vaccines, a lot of information of rumours circulating on social media, rumours in the press, all sorts of things that the COVID vaccines cause. And I think you've had some experience of that in Hong Kong. Would you like to comment on that, Ian?
Yeah, I think the challenge is actually how to use the spontaneous reporting data. Of course, for the people in ISoP, this is our bread and butter. We've been using it for such a long time. And a lot of people don't realize that the so-called causality side of it is a lot of spontaneous reporting systems don't actually say that there's a causality or a causal relationship between the vaccine and the adverse event. And indeed, what you just mentioned is about a paper published in Vaccines, I think about 2 months ago, using the spontaneous reporting data. What the author did wrong was basically he or the group associate the mortality or the death cases as definite caused by the vaccine, and then did some calculation and say that, okay, so every 3 lives that we save using the COVID-19 vaccine, then we kill 2 people because of the vaccine. It was very sensational. It was amazing that within a very short period of time, few days, it's been go haywire in mass media and been download talking about £250,000 of this type of situation. And of course, the paper was redacted very quickly, simply because neither the editor or the reviewer at that point picked it up. The report from the cases from the spontaneous reporting system is not equal to the direct association, and then we see this type of problem occur. And indeed, in early days, because the Hong Kong government was so transparent, so open, so anyone that after they have the COVID-19 vaccine, if they pass away, they will report to the general public. And then the general public misunderstood it. They say, oh no, it's the case must be associated with the vaccine. So it actually hammered the effort of getting people to do the vaccination. So it took a lot of effort to try to make people realize it. Those cases doesn't mean that the people the death caused by the vaccine, but just simply to say that some people die after they have the vaccination.
And I think you said you had to hold some press conferences and information sessions in Hong Kong. And I think that's where those of us in ISoP can hopefully come in and help, is as experts in pharmacovigilance, to actually try and explain causality assessment to the lay public, and to try and demystify some of the rumours that circulate on this causing that. And I know a lot of our ISoP members have got experience in dealing with that, explaining our expert field to people outside that field, so everybody can understand risk-benefit assessments.
Yeah.
So
Mira, can I respond too?
Yes, of course.
Yeah, it's just because you asked the question about the misuse of data and the importance of that from the patient perspective, but you didn't. I just want to highlight that you've got to hold both sides, because the other side is the absence of data and the danger for patients when we have absence of data. So while we're collecting new data, and there's dangers with collecting new data, the harms that are occurring from medicines because of the absence of data are huge. And I think you've got to balance those two. And I think that it's just the conversation needs both bits of that arm.
I think that's an excellent point, and it's also people understanding the type of data and how it's being used is because, as you say, if we don't have denominator data, if we don't if we're not able to do these data linkage studies like Ian and his team have done, then we're not going to get accurate information about the safety of medicine. So yeah, I think that's a really good point, Libby.
Yeah, we are actually running the Hong Kong government is funding us to do another study, not to do the vaccine, but it's actually exploratory study. We recruited children, so teenagers, they have ADHD, and then we give them a Fitbit so that we can use the Fitbit to monitor their heart rate, the movement, the sleep, and everything. And we consent them and we consent the parents, and also we have the assent from them. And then we link up with the hospital authority data. so that we can see how the whole things work. We have a huge amount of linkage data within the healthcare system, but what we tend to missing is like what Libby tried to demonstrate is we are missing the bit about the patient-reported outcome or the outcome that the physiological side of it and the other outcome that we can collect directly from the patient. And we also ask them to fill in the questionnaire about the mood, about the sleeping pattern, so that we can see how the ADHD, patient with ADHD, how the drug can affect those sleeping pattern, the activity pattern, and also when they're missing the dose, what is going to happen. So those are the things that I think for the new generation people, we have amazing response from the new generation. They all love the study. And of course, we also give them a Fitbit. So because of the COVID-19,
that's why they love it.
Yeah, we can't take the Fitbit back because of the COVID-19. So they keep their Fitbit. So initially, we thought we were going to recruit about 5 people for our feasibility study. And we had overwhelming response. So we just expanded to 30, because we thought that would be very difficult to recruit. But the new generation, very different story.
Thanks, Ian. I'm just going to hand back to Joe now and ask are there any questions, Joe, that have come on the chats that we haven't covered?
Yeah, there are.
Or are we running out of time?
Chats. We're probably just about at time, but I wonder if we could just ask, there's one more interesting question for Ian that asks if you have any experience with administration of different vaccines for the same recipient.
Well, in Hong Kong, it's not easy to do that. The reason is because when they have to use the website to book it. So you can't go into one place and then go to another place to have it. The electronic record is so tight. So it's very few people will do that. And so I don't think that we can run the study. However, as you know that the UK already did the study on looking into people who have AstraZeneca and the BioNTech. So I think the study already been published. They did actually say that it's actually a really good response in terms of the antibody, the immunological side of it. But apparently that also slightly increased the adverse drug reaction.
Thank you. Yeah, and my own question was just, did you have a look to see with the Bell's palsy, which side of the face that occurred on and whether there was any association with the injection site, as in which arm the injection was in, or yeah, anything about that?
Yeah, that was one of the initial thinking of most people, to be closer to the injection site. But looking in the data itself, we can't draw any conclusion out of it.
Thank you. It's just after 10 now, Mira. So we did have a few more questions in the chat, but it might be time to draw the session to.
Yeah, I think we've covered most things. And so on behalf of ISoP, I would like to thank everybody for your participation today. Joe for chairing the session with me, and Ian and Libby for your excellent presentations. Thank you so much. I think we've all got a lot out of it, and it's been interesting and stimulating. I would really like to thank Jenny Mercier, and who's part of Angelo Caro's team in ISoP, who've actually worked tirelessly for months to put these webinars together for World Patient Safety Day. And I know what an enormous amount of work it involves, and I really want to thank you personally for all that you do for ISoP. This is it's the opening webinar of the 2021 World Patient Day series for ISoP, and the next session it comes to you from Asia, and I'm told it starts about 12 noon UK time, which I think is about 11 PM in New Zealand, so everyone can work out the timing for them, but do get along to the other sessions if you can. They're going to roll. You've probably seen the posters on social media, and we're covering every region of the world. Every continent is having its own session for World Patient Safety Day in ISoP. And if you're attending this seminar and you are not a member of ISoP, I'd like to warmly invite you to join us. Hopefully you can see that we're a friendly and professional society, and we would really welcome new members who have an interest in pharmacovigilance. Just hop on our website and have a look, and that's isoponline.org, and there's all the details there how to join. And finally, if you'd like to tweet about your experience today, then I don't know whether Jenny can put up the slide as we finish off with the hashtags on. It's this hashtag ISoP medication safety, hashtag World Patient Safety Day. There we go, lovely. So please do share your experience with others on whichever social media you like to use. And I think all that's left for me to do is to thank you all again, and hope I see you all again soon at another ISoP meeting.
Thank you very much. Bye-bye.
Thank you both. Thank you, everybody. Good night.